Hello and welcome to this video on research methods. In this video we're going to look at different types of experiment. So hopefully you're already aware of what an independent and a dependent variable is, what a hypothesis is. You're going to need that pre-knowledge to be able to understand what the differences are between these different types of experiment. So first, it's helpful to define what an experiment is in general. And as I said, good link there to dependent and independent variables. So all experiments have an independent variable, so something that can be changed, and a dependent variable, something that you measure. And then what you would need is some sort of control over those variables. So you may hopefully know some of the strengths and weaknesses of different types of study. And in an experiment, one of the strengths is that we've got what's known as cause and effect. We know A has caused B, or the independent variable has had an effect on the dependent variable. Well, the only way that we know that is if we've got some level of control. So when we talk about different types of study, when we talk about an experiment, there is an IV, there's a DV, and we've got some sort of experimental control. There's also standardized procedure, and again, one of the good strengths of an experiment is that there's replicability. You can repeat your study, or someone else can repeat your study and see if there's the same results there. So we need all of these things in order to have an experiment. We would also need a design, an experimental design. We'll talk about, so this is what we're talking about now is a method, the method that you would use to conduct a study. There's also experimental designs, which is independent measures, repeated measures, how you put your groups and your participants together. Three different types of experiment. There's a laboratory experiment, a field experiment, and a natural experiment. There is actually a further one if you're doing the A level, there's quasi, but this is made for the GCSE. So we're just going to look at those three today. Firstly then, a lab experiment, and this is looking at the AO1 there. So what is a lab experiment? Lab experiments take place in a highly controlled environment. So one of the key differences here between the different types of experiment are where they take place. So in a lab experiment, they are taking place in a very controlled environment. It's not necessarily actually a laboratory, a lab, but it is somewhere where the researcher can really control lots of different variables, and we've already mentioned you need some sort of control. So if you're talking about a study looking at, I don't know, the effect of a type of drink would be the independent variable. You could have tea, coffee, water, and the dependent variable might be attention, and you can measure that, or reaction time. You can measure that in different ways. You do the ruler drop test, for example. You could do that in several different ways. You could ask the participants to come into an environment. Again, it doesn't necessarily have to be a laboratory. It could be somewhere where they could stay for a couple of days. So you can control things like the amount of sleep they've had or the food that they've eaten, because all of those things are things that could affect the DV, could affect attention or reaction time, that are not the IV. So you'd want control over those things. So in an environment where you're getting people to come in, they're staying there, you're controlling all those things, that would be a laboratory environment because it's highly controlled. You would have manipulation of the independent variable. So you would be telling the participants which drink they're having in that example there, and the dependent variable is measured. And I say you can do that in several ways that depend on your operationalization, your definition of your independent and your dependent variable. If you were to do reaction time, for example, you could do there's the ruler drop test, there's online reaction time tests where it can give you a really accurate score. And as I've already said, what that allows you to do is control extraneous or confounding variables. So you would hopefully have a better understanding that your IV is having an effect on DV, and hopefully nothing or not a lot else. There would also be random allocation to conditions. So again, you wouldn't just have the people, I don't know, that would naturally be drinking water and measuring their reaction time, because that could potentially be a confounder. So you as the researcher have got the most amount of control in a laboratory experiment. You would randomly allocate your participants. So you would be telling the participants who's drinking the tea, the water, or the coffee. So in each of these different types of experiment, we've just looked at the lab experiment, that's the AO1. You also need to know the strengths and weaknesses of carrying out these different types of experiments. So there's good things to doing this, but there are also negatives. And so let's look at some AO3. Okay, looking at the evaluations then of lab experiments, the strengths. So there are, of course, good things, and as I said, and hopefully you already got some sort of awareness of the strengths of an experiment in general, but certainly for a lab experiment, you've got good cause and effect there. You know, hopefully, that your IV is the thing that is affecting your DV. You've controlled the extraneous or the confounding variables. So what this gives you is increased what we call validity. So validity is how accurate your results are. So we should have more accurate results in terms of cause and effect with a lab experiment. There's also good standardized procedure, and it is replicable. So what this should do is increase what we call the reliability. So this is how consistently we're likely to get results. If there's lots of things happening and different things happening, confounding variables, things aren't the same each time that this study is run, then you're going to have less consistent results. Whereas if you've got a standardized procedure, if it's replicable, then you're going to have better reliability, so better consistency of results. And finally, again, a good strength of an experiment is that it's meant to be more objective. So that means it's meant to be more factual based. And again, if we've got more control, there's meant to be more objectivity in there. So the results should be more accurate and should be more reliable. However, not all positive, and there are some issues. And as you can imagine, we have what's known as a lack of ecological validity. So this means that are the results really being true? Do the results of the study show what actually happens in real life? So ecological, the environment, validity, accuracy. So does it actually show what happens in the real world? We should be able to do what's known as generalize our results. So we should be able to say, yeah, the results we find here, we can find these time and time again in different samples and in different conditions. Well, if your conditions are really highly controlled, you're probably not going to be as likely to be able to generalize your results to other environments outside of the experiment. And the procedure and task is the same thing as well. So, things happen differently in the real world compared to what, people do eat different food and sleep for different amounts, and that will affect their perception or their reaction time. And so by highly controlling those things, you're not actually experimenting what it's really like to experience those feelings as a human. And that's obviously an issue in psychology where we're testing humans. And finally, we could have known what's known as demand characteristics. So naturally, if you were watched by someone, a bit creepy, but you would naturally act differently than if you were just doing things on your own. And so the very fact that people are in this highly controlled environment, they may act differently in the study than they would do naturally anyway. And so just by being in a controlled laboratory experiment, it could change the way that people act. And again, that would reduce our validity. The results may not be accurate based on that. Next then, we're going to look at the second type of experiment, and that's known as a field experiment. So it doesn't actually take place in an actual field. What it means is that it takes place in a natural environment. So rather than that study that I spoke about looking at different types of drink on reaction time, well, if you did get people into those controlled environment, like I said, you would be able to control more, you'd be able to cut down on extraneous or confounding variables. However, it's a bit artificial. So what a field experiment does is try to put people into a natural environment. And so actually it's more like how what you're trying to study would happen in real life. So if I was studying things like, if I wanted to know about reaction time, well, actually getting them in a very sterile environment doesn't really work. But actually, if I want to see how people worked in an office environment or in a school, it would probably make more sense to actually test them in that environment. And so that's what we're talking about in a field experiment. So when we say field, we mean in the field, in the area, in the environment in which the people normally act in whatever it is that you're actually trying to study. There is still, so some of the strengths of a lab experiment still can be found here. So you can still have an independent variable, you can still manipulate that, so you can still decide which drinks people are having, you can still measure the dependent variable, and there can still be some control. However, it might be limited more limited. It will be more limited than a lab experiment. So there's a bit of a payoff there between being more kind of realistic and in the real environment compared to the control that you might have before. You can also randomly allocate to conditions. So again, that experiment I was talking about with the different types of drink and the reaction time, well, I can still say, okay, I want this group to have water, this group to have tea, this group to have coffee. So I can still have that random allocation. But obviously in a lab, people will have eaten different things. They will have slept for different amounts. That's what I'm losing for not doing it in a lab study, but and doing it in a field study. But in a field study, at least the environment is a bit more realistic, a bit more natural. Okay. So again, strengths and weaknesses to doing field experiments. So firstly, a field experiment has better ecological validity than a lab experiment. So we said the lab experiment, the weakness is that it's a bit artificial, people act differently. Well, in a field experiment, you get a bit more of that, so that's better. And so we can then generalize our findings a bit more. And also, it reduces the demand characteristics. So again, if you've got people in a really controlled environment, you're watching them behind a two-way mirror, which sometimes does happen, they're going to certainly act differently. Whereas if you've got them in their natural school or work environment, then they might act a bit more naturally in relation to that. And so you're going to reduce these demand characteristics. People might act a bit more naturally. However, again, as you can imagine, strength of one is a weakness of another. So you're going to have lost a bit of control. So in the natural environment, there will be probably more extraneous or confounding variables, which will reduce the validity, and it will mean we've got a harder issue, or it becomes harder to establish that cause and effect. We also probably lose a bit of reliability. So if someone were to come and try to repeat the test that I've done, or do it in a different school, then again, those environmental factors are going to be different in different places, so it's harder to replicate and makes the study a bit less reliable, so you might not get those consistent results. And then finally, there are potentially ethical issues here. We'll talk about this a bit later on, but if you're studying people in their own environment, do they know exactly what they're doing? Is the way that you've done the study, do they know you're going to be there? Do they know you're observing all of their behaviour? And if they don't, there's an ethical problem there, and the ethical issue is a lack of informed consent. If you get questions about this in the exam, don't forget to put your answers in context. So use the STEM and relate your strengths and weaknesses to the actual study that's being spoken about. Don't just use the generic responses that are here. So we've just spoken about lab and field experiments. These are together probably known as true experiments. So that's because you directly manipulate the independent variable, you're looking for the change in the independent variable on the dependent variable, and you can randomly allocate to conditions. The next type of experiment is called a natural experiment, and that is slightly different, as you will see. So the natural experiment is different because again, it happens in a natural environment. However, what happens here is that in the previous two, in either the lab or in the workplace, I, as the experimenter, have been the one who's decided who has tea, coffee, or water. I'm trying to measure the effect of what I do, how the manipulation I make on the independent variable, on the dependent variable, on alertness, or whatever it is. You could do that in a different way. So you could do that where you ask, okay, who's had some water, who's had some coffee, and who's had some tea, and use those people as your gauges. So there's again, as you can imagine, there are strengths and weaknesses to doing it that way. The definition here is that the IV varies naturally. So I, as a researcher, haven't decided who's drinking tea, coffee, or water. That has already happened in the environment. I've had less input into the control here. And so that's how a natural experiment differs from a field experiment. There was a good example of this. So there's an island called St Helena, and in 1995 they introduced TV for the first time. So researchers looked at antisocial behaviour on the island before TV was brought in, and after it was brought in. So they, as researchers, didn't decide that the island was going to get TV, but they could still use it as a type of experiment, a natural experiment, to see, okay, does TV have an effect on antisocial behaviour or not? You see there how the ivy naturally varied. And so, yeah, you know if the difference was shown or not. And again, there are strengths and weaknesses here. There will be good things or bad things, and quite as with the other types of experiment, it comes down to that trade-off between how natural the behaviour is and how much control. So you've got more natural behaviour here, and it actually would allow you to study things that would be very difficult to study by manipulating them. So, a researcher is never going to be able to decide that an island has TV and doesn't have TV. So it allowed you to study things that would happen naturally. There have been examples of identical twins or triplets separated by an age. Or, well, actually, psychologists did do that sometimes. But if it happened naturally, then it would be a natural experiment. Whereas if the psychologist did it, then it becomes a field experiment. And so, yeah, ethical issues, it sometimes gets over those ethical issues if this thing is going to be happening anyway and you're just studying it as a psychologist. The ecological validity here is really, really good because these things are naturally occurring. So you can't really get any more natural because these things are happening anyway. You're just going in as the scientist to see what is happening. The problem here is, though, that there's this lack of control, so you don't know what other things have caused the dependent variable to change. So, for example, in that St Helena example, it could just have been that the more antisocial people were more likely to watch TV anyway. And so you could say, okay, well, watching TV makes you more antisocial. Well, actually not. It was just that was going to happen as it is. So you've got a lack of control. There's no allocation condition. It reduces the validity, so how accurate the results are, and also replicability. Those things might not ever happen again. So there's something to be aware of there. So just as a bit of a summary, all experiments have an IV-DV procedure and some sort of control, but they vary in those. So lab experiments, you can manipulate the independent variable in a really controlled but artificial environment. They've got really good control, really good internal validity and reliability, but bad what we know as external validity. Is quite artificial. Field experiments, you manipulate the independent variable in a natural environment, good ecological validity, so the environment's more natural, but your control and therefore your replicability may be reduced. Quasi-experiments are similar to natural. As I say, you don't need to know them unless you're doing the A-level. They're naturally occurring pre-existing, whereas the natural experiments are naturally varying independent variables and tend to happen more in that environment. Of course, they're in that environment because they're happening anyway. Experiments, so that covers all different types of experiment. There are other ways, other methods, other research methods. The topic is called research methods. These other methods are things like case studies or correlations or things like that. That's a bit later, but these are the three different types of experiment you need to know for the GCSE psychology. Thank you for listening.